Biophotonics is the scientific field which is investigating biological systems using light as a probe. It's very intriguing. It's very intriguing because it requires a high-level knowledge in biology, in chemistry, in physics, in optics, in electronics, to build up the tools and the instruments that are needed in order to learn more about every living being. Now, what has to do light with biological system? Well, think about the fireflies. In the fireflies, you have, say, in living animals, production of light by chemical processes. Then you can have, this is called bioluminescence. You can have chemiluminescence, where you are injecting into the object of studies molecules that are interacting with the environment, and as a result of the reactions, you produce light. Or you can have fluorescence, that is to say you have an external source of light which is illuminating what you're studying, and as a result of the interaction between light and the living being, then light can be absorbed, can be scattered, can be diffused. And by looking at the properties of the light, then again, you learn a lot about the processes, the underlying processes in the object of your studies. What do you measure about light? You can measure the intensity. You can measure the fact that light is emitted not in a random way, but synchronized to the emission. You can measure actually the properties by which quanta of light, photons, are emitted. So you do need sophisticated tools to be able to do that. You need tools that are able to see the single quanta of light and to tell not only when it is there, but how often it comes, the rate, the speed, how many photons you have per unit time. You look at your clock, you say how many photons I had per second, per millisecond, per microsecond, per nanosecond. You need to be fast and you need to be good. So in the past, single photon detection was actually something that was obtained using photomultiplier tubes. A fantastic piece of engineering and physics based on the quantum property of matters and the interaction of matters with photons. But they were bulky, they require high voltage, they were even expensive. Today, you do something similar or even better using silicon. We are living in the silicon age. It was somehow quite normal to have this technology moving from an electromechanical object into a piece of silicon. The toys that we use are called silicon photomultipliers. They were born in research, they moved to industry. They are one of the best showcase of Hamamatsu, where photons do matter. What do you do then? You need to be able to blink your eyes when the single photon train comes, to see how often the photon comes. You need to be able to timestamp, and using silicon photomultipliers you can do it with nanosecond resolution or even better than, and then you have to analyze the data. Now here we have an example of the application that my research team work on. So what do? We investigated some very interesting properties of cells. You know, signaling is the method by which cells are interacting with the environment. And signaling can be driven by different elements, among which is it of utmost importance calcium. Calcium ions are really crucial in properties that are defining the well-being of the pathologies related to cells, including the brain works, and then anomalies into calcium-driven signaling can really be an indication of diseases that are occurring. And also the way our heart is working is somehow associated to calcium signaling. So being able to monitor calcium signaling is essential. How do we do it? We do it not using fireflies, we do it using jellyfishes. That looks a bit weird, but there's a protein which is part of the crystal jellyfish, which is quite useful because its photo emission is associated to calcium. So calcium is triggering the emission of lights in the form of series of photons. So together with colleagues and friends, we were setting up an instrument that was monitoring into cells the emission of the photons stimulated by calcium. And what you see here is actually our apparatus. We have one collection of silicon photomultiplier, which is now interfaced to a cell, and into the cells we have cells. We have cells that are suspended in water and are ready to receive an injection of calcium. They were somehow having already aquarine extracted by the jellyfish in, and now injecting calcium, we are able to see how the rate of emission of the photon changes, and then later on to monitor in living cells. And this is what we see. We see here the trace, which is corresponding to the rate of the emission of the photons when no calcium is injected. It blinks. We have 8 channels because we need also to monitor where this is occurring. And now, thanks to the support of one of our young colleagues, we will be actually injecting the calcium in there. It does it, it goes, and there you see really in an impressive way that something is occurring. And this is telling us that the cells that we're looking at properly reactive. This is the beginning of the story. The end of the story will be then to monitor and to develop methods that are able to identify pathologies and learn and produce knowledge, which is then flow back to us, thanks to the technology developed by Hamamatsu.